Good evening. This is the last bit of notes you will take for this semester in Allied Health Biology. Woo! Yay! Cheer, cheer, cheer. Then you're going to have time to study for your exam that you take next week, and you're going to rock that because, like, these guys forgot to make the backup copy of your brain, but I'm sure that every one of you has kept the information and it was not lost at all. Okay? So in your notes, we are at the point where it says a more detailed look at translation. I know we've spoke about translation, but here we're going to get a few more details with it. Okay, so the ribosome, it first of all says, is it's made up of two subunits. Our subunits are here with the large part and here with the small part. This word proteins, I apologize. When I make the recording, the words don't exactly go on the lines, and I haven't figured out how to fix that. So just kind of have to deal with some of those blanks there. Ribosomes are made in the nucleolus. We learned that in chapter 7. And so the ribosomes themselves made in the nucleolus, then they go to the cytoplasm where they wait for the RNA to tell them what to do. Okay, so in this picture, what we're looking at, and everything's pointed out to you, but okay, so we have the large subunit and the small subunit. This whole thing all together is our messenger RNA. Okay, and you see this transfer RNA right here is holding the growing protein. So all these things here are showing how the protein is growing because of the amino acids that keep getting delivered there. Okay, so this transfer RNA right here is delivering the next amino acid for the protein. So this little diamond is going to be added here. Okay, and then this transfer RNA, it's done, and so it's going to be leaving that ribosome and going back into the cytoplasm there. Okay, so the building of the protein. Transfer RNA begins at AUG. Remember, that's the start codon, and that's always going to be the start codon. Each transfer RNA has an anticodon. Remember, that's at the very bottom of it, and the bases are complementary. Complementary bases means that, of course, in the RNA, A's will pair with U's, C's will pair with G's, and vice versa. And it's important that we know when we do this decoding and such that it is the messenger RNA that is telling us which amino acid it is. Okay, so in this case, our AUG is on the messenger RNA, and if we look at our table in page 303, that tells us that that is methionine. And so that's always the start codon. That's where it's always going to begin the process of protein synthesis. So we hit here, and this diagram, it is very similar to the others, but with a little bit of explanation now. So the first transfer RNA has completed its job. Okay, so I'm right here, and it's going to exit. Okay, and it leaves the cytoplasm. It leaves the ribosome and goes into the cytoplasm. So another transfer RNA, that's this one here, arrives with the next amino acid, and it's going to kind of connect right there, and you'll see that in this picture here. It shifts the growing polypeptide, that's these things, and we get new transfer RNAs coming on. The ribosome forms a bond. This is actually a peptide bond with the amino acids, and they keep shifting over, and you'll see that this chain keeps growing. Okay, so then we get that they're added one by one until we get the stop codon. Okay, so all of these, we're going to add these different shapes for the protein up until we get a stop codon. Remember, there are 3 of those, and the end result, we finally get to a protein. Okay, so here, the big question that I have right here for us is, what do proteins have to do with the color of a flower, human blood type, or dimples? And of course, right there in the middle of the page, you see it is everything. Proteins being built within the cells. That's what the traits are a result of. Okay, so the genes, basically, giving us all those traits that we've learned about, are instructions for building proteins. Mutations. This can happen. We oftentimes think of mutations as being very, very bad, and we get the Hollywood and such like that, or Teenage Mutant Ninja Turtles and such, but that's not always the case, okay? A mutation is simply a mistake in copying the DNA, An incorrect nitrogen base may be inserted or it can be skipped altogether. These are called mutations. Even though we've got the polymerase, which does the checking of it, mistakes can be made. Mutations important. It can be on the gene, which is just a small little piece of the chromosome, or it can be a whole chromosome mutation. We've got both. And the chromosome mutations are usually a little bit more prevalent, or we see them more, or a little bit more harmful. Okay. And there's several different types of mutations. Here we get a point mutation. And if we look at in this messenger RNA here, okay, we see that it should have been copied as GAA, and that would give us normal hemoglobin. Hemoglobin refers to blood. Hemo is blood. Okay, but this is copied incorrectly. Instead of an A, we have a U. This is where we get sickle cell. Okay, and that's a point mutation. It's just one small thing that is incorrect. It didn't affect the whole sequence of DNA, just that one part. And this is what it causes. Sickle cell anemia is a disease that affects the red blood cells in people, and they're born with it. There is no cure because it is on their genes. The normal red blood cells are nice and circular like this. Sickle cell, you see, looks more like a football. And what happens with sickle cell is these actually kind of get caught in the veins and arteries as the blood is flowing. And, you know, if something blocks that, the blood isn't going to flow well. And if that happens, then the organs aren't going to get the oxygen they need. It can be very, very painful for somebody with sickle cell anemia. It can also kill them because they can get blood clots very easily and end up getting strokes and heart attacks very easily and at very young ages. Okay, another type is this base pair substitution. What I mean by substitution, a substitution, think of a teacher when she's got or he has to be out for a day, you get a substitute. So it's replacing a nitrogen base with another. Okay, so here we see that happening here. And sorry for these words that are out of whack and such, but that happened when I copied it over. So in this blank, it should say no effect. And this would call for alanine. These are silent mutations, meaning we really don't know about them. In our bodies, we have many, many mutations that we don't even know about because remember, it's just a change in that DNA. Substitution, we're in the first or second position, it would have a greater effect. Switched amino acid may or may not have an effect on the properly functioning of that. Okay, here's these questions. What amino acid would be called for by the codon AAG? You can look that up on page 303 of your book. So hit pause if you need to look in your book to answer these, because I wouldn't expect you to memorize this. What would happen if an A was substituted at the third position in this codon? So an A would be substituted here. What's going to happen? Check out page 303 and see what that changes to. What would happen if a C was substituted at the third in this? So now instead of G, you have C. What would happen if there was a substitution at the first or second position in this codon? Okay, push pause, check page 303 in your book, and answer these questions. Okay, now here are your answers. So AAG is lysine. If an A is substituted, it's still lysine. No big deal. So we have no effect on the protein. But if we have AAC, now that's asparagine, and we have that protein being altered. Okay, so insertions and deletions. We can add or delete a nitrogen base. This would have a huge effect on it. Codons are still read in groups of 3. Properly grouped in codons. These are frame shift mutations. Excuse me there. The importance. Most gene mutations are neutral. They have little or no effect. Some can cause dramatic changes. Genetic disorders come by this way. And I do have many examples of those. So just because we finish the notes, please stay tuned for some examples of this. Some can be beneficial. Looking at these birds, these beaks on these birds, these are all mutations. And if the birds were actually able to get different food that might be in that environment and help them survive better, that's a beneficial mutation. Okay, so most are neutral. Neutrals, mutations might be eye color or birthmarks. Yes, that is a mutation. For those of you that have a birthmark, you know you've got a mutation. Your eye color, it's hard to tell whether your particular eye color is a mutation or not, but it could be. Harmful mutations would be sickle cell anemia and Down syndrome. I've got a picture of Down syndrome in a second. But beneficial, sickle cell anemia to malaria. People who have sickle cell anemia, now the majority of people affected by this disease live in Africa. In Africa, there are a lot of mosquitoes, and mosquitoes carry malaria. The nice thing is, those people who are affected by sickle cell do not tend to get malaria. And so they are immune to that. And there are some people who are actually immune to HIV due to a mutation. HIV is the virus that leads to AIDS. Okay, causes, they can be inherited. A parent has it, they pass it down to their child. This is only going to be if the mutation is actually in the sperm or the egg to pass down. Okay, and it can be acquired. Environmental, this would be like smoking, pollution, things like that, cancerous substances, or make a mistake when the DNA is copied. Here's some chromosome mutations. Down syndrome. She's beautiful. Okay, Down syndrome people are probably the nicest people you will ever meet. They're sweet, always smiling, and they are extremely strong. Some of them don't even know how strong they are. But it happens because chromosome 21 does not separate correctly. We often call this trisomy 21. And we get cry to cat. Yeah, I know it says chat, but it's actually pronounced cat because these babies, when they cry, it sounds like a cat. And they'd have various mental handicaps. So I know it says mental. It should say mental. I apologize for that. Klinefelters. Okay, you'll see with Klinefelters, it always has this Y, so it's got to be a boy, but they have extra X's. They're generally sterile, small testicles, breast enlargement. A lot of these people do end up in jail, unfortunately, because they end up being very aggressive. Sex chromosome, here we have double Ys as well. This is really more associated with antisocial and behavioral problems. Okay. Turner syndrome, 1X. Remember, you're supposed to get an X from mom and dad if you're female. And so these people would have Turners if they only had the 1X. And their sex organs aren't mature. They're generally sterile and pretty short. Okay. Trisomy X, really not going to be able to tell on these people if they have it. But later on in life, if they're limited fertility, if they're trying to have a baby and can't, that's when they might find out that they have 3 X's. Okay, gigantism. Yes, actual picture from Barnum and Bailey Circus. And gigantism is actually a genetic defect. And I just thought this was fun. Okay, so we are finished with the notes. I wish you all the best on your finals. Study hard. If you have questions, email me or anything like that. I'd be more than happy to help you.